So, just a little bit about my perspective before we get rolling. Uh, we've had continuous glucose sensors. Everybody's used to them. A lot of people have tried them. We have wearables where they're on the wrist and the ring. But what you have come up with is a whole new world of sensors. And this is actually pretty extraordinary because of your fusion of biomedical engineering and chemistry expertise. You've come into something of being able to basically real time, continuous monitor any protein in the body. Wow. Okay. So we're back to you. We got everybody on board. We have our audio fixed. Thank goodness. And maybe you can restart with telling us about how this protein sensor works.
Yeah. Well, I really appreciate, Eric, the way you summed it up. It's something we're very excited about too. And you're right that this all started with glucose sensing, right? It used to be if you were diabetic, you had to go to the pharmacy to get your glucose measured. And then you got a handheld, right? And that really transformed diabetes management. Now we have continuous glucose monitors that sit on the arm and can read glucose in real time. But at the core of that is a sensor, right? That reads out the glucose concentration. And what's been very elusive when it comes to kind of all other analytes, especially protein analytes, is how to get that same form factor. How to have an autonomous sensor that can just sit in a fluid in the human body and read out concentrations. And so that's the problem we've been working away on for several years, many years. And we eventually came up with a sensor that's basically like a molecular motion detector. It sits on the surface of an electrode and it's just it's moving around. We use electric fields to kind of move it in a way that we can monitor. And it turns out that when the sensor binds a protein of interest, that slows it down. And so we can just watch that motion, do that over time, and quantitate concentrations of proteins in the blood or in the interstitial fluid is usually where we're making our measurements.
Yeah. So it's like a molecular pendulum where you have these strands of DNA that are specifically want to bind to a protein, but it binds so well that you need a little electricity to shake it off. Is that right?
Yeah, that's exactly right. So I have a puppy at home and we throw balls for him all the time. He's really good. He'll go get exactly the ball that you just threw. He'll bring it back, but then he won't let it go.
Yeah,
Same thing. So
every
the
do-
sensor catches the protein, but it won't let it go. It's a very strong complex.
yeah, every dog I've had the same problem. Never let the ball go, and that's kind of like the metaphor's powerful. So you figure out how to do this, which is with no reagents. This is unlike a continuous glucose sensor or a lactate sensor. No reagents, a tiny bit of electricity, and then you sense the protein level. And then you go ahead and you say, okay, let's try this in a diabetic rat model. And you're looking for specific inflammation proteins like interleukin 6, tumor necrosis factor. And what did you find when you did that?
Yeah, so we chose a diabetes model because, diabetes and inflammation go hand in hand, right? If your glucose is out of range, your inflammation is going to start to spike. So we thought that would be a good model. And, we had rats that we allowed to fast, and we could see that as they fasted, their pro inflammatory cytokines would come down. We had animals that we would inject with insulin, and we could see the pro inflammatory markers come down even faster. Interestingly, when we injected the rats with insulin to kind of see how that would affect things, we actually saw a little spike in inflammation just from the needle prick. Like that's how good the resolution is. So that was very cool to
It,
see.
I was struck by that, especially. Yeah, yeah.
Yeah. And then we also dosed the rats with molecules that would make their inflammation spike. And we could absolutely see that. So we were able to convince ourselves and the folks that reviewed our paper that we really could watch inflammation in real time in a living animal.
Yeah, so this is why it was such a landmark contribution because no one had ever really conceived that this could be possible. But you actually demonstrated it is more than possible. You demonstrated it in a very impressive animal model. Now, there seems to be limitless clinical applications of this. I mean, so, uh, if you were trying to monitor if a person's eating an anti-inflammatory diet, or they're exercising and they're keeping their body in their low levels of inflammation, which we know inflammaging is such a big part of how we wind up with these age-related diseases. This seems like a perfect way in the future that we could assure that a person's inflammatory state, and it doesn't have to be these proteins, it could be pretty much any protein, right? You could do that in real time. Now, do you see a way where you could get this signal to your smartphone?
Oh, absolutely. I mean, the concept is to just we're gonna just go right alongside, the way the CGMs have been developed. So sensor in the interstitial fluid, app on a phone, being able to track markers that will help us make lifestyle decisions, help us manage disease. And then there's also a whole set of research applications as well that I think will be really interesting.
Yeah, I mean, so for me, so into the new era of prevention that's been potentiated with these markers that you're adding a whole different level of sophistication of marker, I would say. And we don't have very good systemic blood markers now. We use high sensitivity CRP, maybe IL-6, but this is a whole different look, being able to do this in continuous in real time. Now, other ways that this could be used are, for trying to prevent Alzheimer's disease, um, autoimmune diseases. One of the ones that you posited was heart failure by, for example, continuous measurement of uh, the BNP peptides that tell us about the impending heart failure. You may want to comment about this whole idea of using a sensor for protein monitoring for precision medicine.
Yeah, absolutely. You described it very well that for the chronic diseases that are hard to manage, right? Heart failure is a classic one. You know this better than me that you have patients in various stages of heart failure and they may be okay and they seem to be, at a steady state. But then something happens and they wake up and their ankles are swollen and it's not clear what's going on. Do medications need to be titrated? If you could see what BNP had been doing over the last 24 to 48 hours, that would probably give you an answer in terms of what action to take next. So I think chronic disease management is absolutely a place that we can just do so much more for patients if we're able to monitor them. Septic patients in the ICU, right? Really understanding what inflammation, how it's trending there. And then you mentioned neurodegenerative disease. I mean, one of the things that I think is really fascinating to think about as an application, maybe not in humans, but in our model organisms, just really understanding neuroinflammation and
Oh,
what happens in the brain, when you have a spike in inflammation, right? There are people that are diagnosed as being schizophrenic, but they actually just have inflammation in their brains. And when they're treated appropriately, right, the psychosis goes away. But it's very much a black box. So I think we can do a lot for patients if we can take the technology where I would like to, but we can also answer a lot of questions about how inflammation affects us systemically, but also at the organ level.
absolutely. There's unparalleled ways that this can go forward in both, as you say, research, but also in the management of and prevention of diseases. I mean, the autoimmune disease category is way up there in the list. Now, how do you move forward? You got this ingenious invention, you've got a proof of it in an animal model. How do we accelerate this so it becomes something that we could use in the clinic?
Yeah, so that's actually the hard part. I mean, the inventing and the proof of concept and all that was hard, and it took us many years to get there. But now turning this technology into a product, that's a whole other exercise. I've run academic labs, but I've also run companies in the diagnostic space, startups that emerge from my labs. I know the product development, roadmaps, the life cycles, and all of this. And that is really what we need to do next. What we're doing right now is just making absolutely sure that we have a very robust prototype. We are developing a prototype for human use so that we can, when we start walking down that commercial path, we really know that we've got something robust in hand to start with. That work will be done during 2026, and then we're ready to take that next step, either with a commercial partner, the Abbotts, Dexcoms, those, players in the field, or, we'll go it our own and have a startup company that takes the next steps with it. But I'm very committed to this. I really believe in it just in the way that you, mentioned that this really could be transformative for the way so many diseases are managed, and so we absolutely wanna see that through.
Yeah, I mean, I've over the years tried to anticipate where things are going, and I haven't been as excited about a new technology as this one, Shana. Now, I want to sign up as patient zero when you're ready. Okay, will you let me
You
do?
got it.
I want to know my inflammation status in real time. All right, now, this is a part of something that is really extraordinary, which is your Chan Zuckerberg initiative biohub. There's only a few of these biohubs in the country, and back in 2023, you were selected in a competition of many cities that wanted to tap into the support of CZI to do this. So, can you tell us about the biohub and how it helps enable this type of work?
Yeah, absolutely. So the biohubs are really unique types of biomedical research institutes. They function in partnership with top tier world class research intensive universities. In Chicago, we work with the University of Chicago, Northwestern, University of Illinois, Urbana Champaign. And we're really here to do high risk, high reward research, to really be out in white space where there isn't a lot of activity, in, let's say, the NIH funded parts of biomedical research. And what I think is really cool about the model is that we bring together scientists, chemists, physicists, biologists, with engineers, with folks that do AI. It's very multidisciplinary. And we assemble these incredible teams to work away on hard problems, like the problems that we've been talking about: sensing inflammation, watching it in real time, profiling inflammation, understanding autoimmune disease. That's our focus at the Biohub in Chicago. And, I run an NIH funded lab for 20 years, and it's wonderful to have that kind of support. But the support that we get at the Biohub allows us to move fast, fail fast too, if we're going to, but, really move fast towards problems that will have significant impact.
Yeah, well, it's great that you get that added support, especially where at a time when it's hard to count on NIH the way things are headed or going. But also, there's another force there, a major force besides your team, which is another pioneer in sensors, John Rogers, who he was at U of I, which I guess is also part of your consortium. But he's more on the physiology sensors. Obviously, so I look at what's going on in Northwestern, and I mean, I may be off on this, but it seems to be the capital of the world of sensors, futuristic sensors, and yours is, one I think that is unparalleled. I don't know anyone else who's been working on, the ability to track proteins. It could be tracking DNA, RNA, things that we've not been able to do in the past. And whereas the work that John's been doing, as I understand it, is largely physiology centered. Is that a good way to summarize it?
That's correct. Yeah. John is a wonderful colleague at Northwestern and a bioelectronics, extraordinaire. His team builds these amazing devices, a lot of flexible devices that can go on the skin. He's done a lot of work on devices for neonatal care and, just delivering the kind of monitoring capability that you would want for babies in a NICU. So, yeah, we're more on the molecular side and he's more on the physiological side. It's all very complementary. But that was one of the reasons I found Northwestern to be a really attractive place to do my work, because we are extraordinary in bioelectronics.
Well, some people might know Chicago for deep dish pizza, but I know it for sensor pioneering work. Yeah, and by the way, if you have questions that you want to ask Shana, please put them in the message, and we'll try to make sure that we respond to them as we're talking. Now, besides the consortium you have, that is taking on this work, you mentioned something that's really important is the AI side of it. So, one of the most difficult things in AI is the analysis of data from continuous sensors. Uh, the words that are used are time series analysis. So in order, there's so much data that's generated. People think the genome has got a lot of data. Well, you have a continuous sensors going on, and you're trying to analyze that with all the other layers of a person data. So, have you been working on how to do that type of analytic work? Because that's another part of the challenge.
Absolutely. So, really, everything we do at the Biohub, there's an AI element that is either accelerating it or driving it. And we see sensor data. I mean, a continuous stream of molecular data is something that will be very powerful for the development of models. And I think as we take this technology and we scale it and we multiplex it, and we're, right now looking at 2 things, but then we're looking at 10 things, we're looking at 50 things over a time scale of weeks, right? This is an incredibly complex data set. And, my dream, I'm sure you've seen these charts of, like, the metabolic everything in the human body, right? The Krebs cycle and all of this different stuff. But it's all kind of textbooky, right? In the human body, things are a lot more complicated. And with sensors, right, if we have the ability to have this real time data, that complicated scheme really comes to life in terms of what it means for human physiology. But I do think that having the computational models really driving that will be essential.
Yeah, I couldn't agree with you more. I mean, the ability to take the analytics to inform an individual about, that they could be having impending heart failure, that they're having high levels of inflammation, that something is off track, to have that do that reliably so they're not getting false signals. It's another part of the challenge beyond what you've already done, which is really quite extraordinary. Now, what's also amazing about you is that this is only one of the dimensions of your work. I mean, I was looking at your recent publications, and it is, you've got all this work on rare cells, what lymphocytes and macrophages. You've got, many different aspects of what you're doing that are apart from this. So maybe you can just fill us in that maybe this is the principal part that's got you most excited, but you're using, because you have molecular probes, um, in the live person, or at this point, perhaps you say models, you're going in directions for intracellular delivery. I mean, wow. Tell us more about these other things that you're into now.
Sure. So my labs over the years were really not the type of folks that just work on one problem. We're really technology developers. I was trained as a chemist at Caltech, not the molecule making kind of chemist, but like the molecule measuring kind of chemist. And we really just, we look for white space, we look for unsolved problems, unmet needs. And then, the beginning of our project is always standing at a whiteboard and saying, how can we measure this? And if we
Yeah.
could measure this, what could we do with it? So we started a project a few years ago where we wanted a way to be able to search for rare cells in the blood much faster. You can use a flow cytometer, but to get through a milliliter of blood is going to take you weeks and weeks and weeks. The cells will be dead by then. So we developed a high throughput technology for cellular analysis, and we ended up being able to find immune cells in the blood that had been inside of a tumor and had come out. So these are tumor reactive lymphocytes. And what we learned is that we could isolate enough of those that we could treat a tumor with them. So this is just kind of how the road of serendipity usually goes for us. Unmet need, technology platform, always takes a while to find that kind of really compelling application, but we work and work and work till we find something, and then we partner, we spin companies out. That company in particular that is taking the bloodborne tumor reactive lymphocytes is taking that approach towards the clinic, and that's really my passion is developing these things and then trying to move them along so that they can have a positive impact on patients. And we're just kind of agnostic about that. All that really matters is the impact.
Yeah, I mean, that one is striking because if you can monitor a person that you're trying to treat their cancer, successfully, and know that you're doing well with respect to, this whole tumor infiltrating lymphocytes. If you can know what's going on in those dynamics, know
Mhm.
that point about you could take these cells and use them to be part of the treatment. It's pretty striking. And what about this intracellular delivery thing? That's another zone that I was unclear. I didn't know you were working on that too.
Yeah, that again, it was just another unsolved problem, something that we spotted that we thought was really interesting, and the problem that we're trying to tackle there is how do you get a molecule into a cell but then into the mitochondria of the cell, right? The mitochondria are doing all these really interesting things. Those are the energy producers, obviously, but they have a lot of other signaling capabilities. There's DNA in mitochondria, which we don't completely understand what it does or doesn't do. So we just started developing chemical probes that could get there, and then we could ask a lot of questions about how all of the machinery in mitochondria works together. And it's been really fascinating. No startup there yet, but it's been a really interesting project.
Yeah, yeah, no. So I just wanted to make sure that people who are listening, watching, realize that even though this ingenious work on the continuous sensor of proteins, it's just one part of a multifaceted approach that you're taking that are pretty unique, pretty impressive. Now, let's go back to the principal topic, which is the ability to track a protein or proteins in a person on a continuous basis in real time, reporting to that individual if there's something that's off track. This came about, it seems to me, in part because of interdisciplinary expertise. You have yourself a chemistry and biomedical engineering background at the leading institutions in the country. So you think of things differently, and then you probably have a team that does that. You have people coming from different I mean, when you cracked this case, which, was only something you could imagine some years ago, do you think that this was the key that you just because you have a fusion of different areas of biomedical uh, expertise?
Yeah, I think, being interdisciplinary really helps. You have to bring different things together. There were also some very, very talented engineers and chemists on the team that could make it happen and had the skills to put it all together. But I think it's also, being in the space for a while, also straddling the research world and the commercial world and kind of knowing what the unmet needs are in the industry and just understanding those big problems. And I think from what I've said, I'm just kind of a problem person. I'm not somebody who's like a basic science. I have incredible respect for people that just go super deep with a basic science problem. But for me, it's much more about the application and what is impossible right now that we could make possible. And going after those kinds of problems, to me, that's why being a scientist is just so awesome. I mean, I love this path of discovery and then attacking, hard problems that could have a major impact.
Well, it certainly shows. Now, are you aware of any other group around the world that has done work like this where, you know, without reagents, little electricity, shaking off the protein to be able to assay it? I mean, this is something that I haven't heard elsewhere besides your. Is there any other group that's onto it now?
Yeah, we have been the group that's been most active on protein sensing. There is also some wonderful work on developing sensors for other small molecules, kind of, way beyond glucose. So there's Kevin Plasko at UCSB, there's Tom So at Stanford, Jason Heikenfeld in Cincinnati, a lot of people that are taking sensors for small molecules, using them to look at drugs being metabolized in the bloodstream, looking at various things that will also, I think, be really powerful as clinical tools. So we've just stayed over on the protein side of things because I just historically, have looked at chronic disease management and how difficult it is and the need for other tools. But we're certainly not alone in terms of a keen interest in developing electrochemical sensors and really believing that like the CGM like paradigm is the way we're going to get there for
There's
more in-depth monitoring.
is a good way to put it. And we're talking about nanoscale, nanotechnology. And a lot of people have been wondering, when is nanotechnology going to change my life and my health? It seems like you've been doing that. Now we're also in a very unique era where um, this kind of large language of life models, so many things that are fulfilling this the kind of digital biology or biology becoming an engineering discipline in many respects, and your work exemplifies that. So things are really taking off in terms of this, not just the language of life, but being able to understand a person's life and health on a basis that we probably might not have thought about. A few years ago, maybe even a few months ago. So where do we go from here? I guess just to sum up, you're pursuing the clinical path for the protein sensor. Maybe you'll be able to get into people in a year and a half, perhaps. Is that
Yep.
fair?
That's what we're aiming for.
Right. And I'm signing myself up to be the first
This is
one.
being recorded, Eric, so we'll take
Yeah,
you up on that.
that'd be, that'd be exciting. Yeah, what you're doing is really extraordinary. I just want to congratulate you on that. And if there's any questions before we wrap up, this is a good time to put them in the message. Somebody did ask about colchicine, like for example, if you have atherosclerosis and you had inflammation in your denoting artery inflammation, would you be able to see whether the medications are bringing down the inflammation? You would agree that's probably a good way to gauge the ability to suppress inflammation.
Yeah, absolutely. I mean, right now we are focused on systemic inflammation. So if whatever markers, give you the right output end up in the interstitial fluid, we can get at that. But we are also looking at developing devices that, for example, could be implanted in the heart to be closer to the source of the problem so that we can get at organ level inflammation rather than kind of looking at everything going on in the body. So I think the adaptability is there. I'm always amazed, when I talk to cardiologists. Seems like there's a lot you can do with the heart and devices. There's a lot that's been done already. So I think that's a very interesting potential future application.
Yeah, no, I think you probably could pick up what's going on in the arteries without even, just from the sensor you had already put in the animal model, and see whether you're suppressing inflammation. Another person asked about long COVID, and that's really intriguing because here you have dysregulated uh, immune system, you have often high levels of inflammation, and then we don't have a treatment. So this might be a really good way, a validated treatment, to see whether or not you're making progress in long COVID. We don't even have a biomarker, but it could be your sensor is a way to really light that area up. Don't you think?
I do think so. I mean, anything that has that kind of black box kind of feature to it, right? Where we just really don't understand the drivers. People feel better on a given day. They don't know why. They feel worse on another day. If we had the biochemical data, we could probably deconvolute that. Same thing with the autoimmune diseases that kind of have these spikes and people are trying to modify diet or lifestyle in different ways, but it's just really hard to understand what the levers are. Having biochemical data would make a huge difference.
All right. Well, we touched on a few of the questions, which are really good ones. I also want to just say, uh, gotten a glimpse to the future here, folks. Those of you who are here with us live or listening or watching the archive. The reason is because this is giving us a new opportunity for individual that is promoting the health of an individual. So it's really pretty extraordinary. Um, you may not know it today, and you may start seeing results in people in 2027. But what you started here is just so extraordinary. So, Shana, I just want to say again, what you've done, consider ingenious. I think now, hopefully, people will know Northwestern and Chicago. and the efforts that are being supported there by CZI is something that is just to watch carefully. There's a lot of great stuff gonna happen out of it. And so, hope you'll keep us posted. I do wanna, say there's another really impressive review coming out on this topic by Professor Kelley and her colleagues, so stay tuned for that later this month. And it's just the beginning of a revolution occurring in medicine that adds on to what we have today. It's really so darn impressive. So thanks for joining. We're sorry for the technical problems on the beginning with audio, but I think we finally got that resolved. And we've had several hundred people join us, and I'm sure thousands more will be watching subsequently. So, Shana, thank you. And thanks again for bearing with the issues that we had technically to get started.
My pleasure. Thank you so much, Eric.
All right. Take care.
Okay, bye-bye.